I'm Philip Garcia, and I'm going to be giving my presentation, my video over cells. I will be discussing both prokaryotes and eukaryotes. However, the one I'm going to start off with are eukaryotes, specifically an animal cell. Now I numbered each of the organelles that are important in a eukaryote, and as you will see later, I did the same with the prokaryotes. I number them. The important parts of the eukaryotes, which I will be talking about today. Now to start off with, I want to start with the nucleosis. The nucleosis is involved in ribosome synthesis. A nucleus can have several nuclei, so several of those. Now the next one, number 2, is the chromatin, and that consists of the DNA proteins seen as condensed, and it is seen as condensed chromosomes during cell division. Next is the nuclear envelope. The nuclear envelope's job is to protect the DNA and the chromatin from what's going on within the cell. Next is the entire nucleus. It's pretty much all these parts I've talked about, 1, 2, and 3. 4 makes up all those three parts. But it houses the DNA. It's a site for DNA replication, transcription, and ribosome synthesis. Next, I'm going to talk about the rough ER, and that's involved in protein and phospholipid synthesis by transporting them to the Golgi apparatus in vesicles. They have ribosomes attached. As you can see, those little brown spots all around it. That's how you can distinguish the rough ER between the smooth ERs that we'll get to next. The smooth ER synthesizes lipids and regulates calcium levels. It also breaks down toxic substances. So it's kind of like a cleaning mechanism for the body, and when it senses something that's out of place. The next one I will be talking about is the centrosome, which is right there, the yellow one, in between the mitochondria, and that organizes microtubules and provides structures to the cells. This pulls chromatids apart during cell division as well. Next is mitochondria, the orange 1 and 8. It's a place where cellular respiration occurs and where ATP is mostly generated. Next, the Golgi apparatus, as we mentioned earlier. It modifies, sorts, and secretes cell products. And it's active in synthesis, as we said earlier, as it receives these vesicles from the rough ER. Next is the peroxisome. It has various specialized metabolic functions and produces hydrogen as byproduct and converts it to water. Next are the intermediate filaments, which are these right here, and they provide mechanical support for the plasma membrane, and they don't really participate in the cell motility. The microfilaments are flexible but strong, but resist crushing and buckling while providing the cell with support. Next, I'm going to talk about the microvilli. It increases the cell surface area, pretty much. These little things are called the microvilli. The lysosome, which is right here, is a digestive organelle where micromolecules are hydrolyzed. The plasma membrane is pretty much what encloses the cell, and it encloses the cell's interior and is made of a phospholipid bilayer. Microtubules are these little yellow things coming out of the centrosome, and they form part of the cytoskeleton and provide the cytoplasm with structure and shape. The ribosomes, as we mentioned earlier, are the little brown spots all around the cell, and they make proteins and are found free either in the cytosol or bound to the rough ER or the nuclear envelope. The cytoplasm is pretty much all this yellow stuff, and it's a jelly-like fluid filling the cell and contains all the organelles. Now, this isn't part of the animal cell, but it's part of a plant cell, and it's really important because it's a place where photosynthesis takes place in plant cells. So now moving on to the prokaryotes. I'll get this out of the way. Prokaryote cell. And here, these are my flashcards for this one. The one right there is pretty much the cell's DNA, only it's not enclosed in the nucleus like the eukaryotes is. I'm sorry. The nucleoid is the area where the DNA is located. So pretty much all of this. And the difference between the bacterial chromosome and the nucleoid is bacterial chromosome is pretty much already the DNA, and while the nucleoid is pretty much like the general area where all that's located. Ribosomes, we mentioned this in the last one, but it synthesizes proteins. As you can see, these little blue spots. They're different colors. The color doesn't really matter. The cell wall is a little yellow thing right here, and it's a rigid structure outside the plasma membrane that pretty much encloses stuff from coming in and out of the cell. It reinforces the plasma membrane, and encloses the cytoplasm. The capsule is a 6, the next thing I'll talk about, and it's a jelly-like outer coating of many prokaryotes. And these three enclosures pretty much reinforce each other to keep stuff in and out of the cell. Now, the flagellum is right here. It's a locomotion organelle of some bacteria, so it really helps in the motility of the bacteria. The cytoplasm holds a material within the cell in this jelly-like substance. So it's pretty much all this blue stuff, as we said before. And we saw before it was yellow that time, but this time it's blue. Now the next one is this 10. It's plasmid. It's a small DNA structure that's separated from the rest of the chromosomal DNA. Chromosomal DNA, yeah. Now, fimbriae are these little green hair-like substances that we see, and they're structures that pretty much have a similar effect on flagella, and they help the prokaryote move around. Now, finally, I'm gonna talk about the food granule. It's right there. It's pretty much a secretory vesicle. Secretory vesicle. Yeah. So that's it. Thank you.